Okay, hello everyone. Welcome again to another Genomics Aotearoa seminar. This one, I was going to say it's Friday. It's Friday, and of course we're having a Genomics Aotearoa seminar. Thank you for coming along. If you are not on our mailing list and you'd like to be, please send us stuff. An email to genomics.aotearoa.otago.ac.nz, which is in the chat at the moment. We'll get you on our mailing list and we'll make sure that you get to come to these seminars. Today's seminar is from Eddy Dowle. So Eddy is a postdoctoral researcher, or actually more than a postdoctoral researcher here at the University of Otago. She started her PhD, she did a PhD at Massey University in Palmerston North, and then moved on to fantastic jobs at Kansas State University and the University of Colorado before coming back here, and has a Marsden grant looking at the way that parasites manipulate their hosts, which I think is one of the most fascinating subjects from Marsden I've heard of for a long time. So Eddy's going to be talking to us today about flies, which instantly means it's going to be fantastically interesting. So I'm going to ask her to start up her talk. In the meantime, if you can mute your microphones, turn off your video, just to make sure there's no distractions. If you've got any questions, please throw them into the chat, or we'll have hopefully some time for questions at the end of the talk. So Eddy, you're in charge, off you go.
So kia ora koutou, I'm filling in for someone this week, and as such, this presentation has only got a week's notice, and there's been conferences and teaching getting in the way of me writing a new presentation. So it's pretty much two short presentations stuck together, one of which was aimed at a genetic audience and the other was aimed at a more general audience. So if you want to hear about the actual science details on the later project, you'll have to check back later, as I didn't have much time to think about what I was going to present. I also grabbed an old title, but it's only actually half the talk. The other half is going to be about parasites, which Peter mentioned. So maybe a better title would have been something like this. But I thought I'd also give you an overview of who I am and what I do as well. So obviously at the moment I work on using transcriptomics to look at insect evolutionary development biology, but it's not what my original background was. So I started off, although I'm originally from Otago, I started my research at Massey University in Palmerston North, and I was lucky enough to work with Steve Truick and Mary Morgan Richards. And I owe them a lot of thanks for getting me into genetics and showing me the light that invertebrates are pretty cool to work with. And when I was there, I did my honours on some grasshopper work, and I did my PhD on some Weta and New Caledonian Placostylus snails. When I finished my PhD, I actually moved to Nelson for a while, where I worked at the Cawthron Institute with some pretty neat folks down there. And when I was working at the Cawthron Institute, we were looking at developing genomic approaches for macroinvertebrate biomonitoring largely. And then I made the really strange decision to move to Kansas in the States. So if you're not geographically minded, Kansas is in the center of the States, and it's flat land associated with sunflowers and the Wizard of Oz. And obviously, as much as I like the Wizard of Oz, I didn't actually move there for that. I did move there to do some cool science in the Gemmell Lab. And for most of the time in the States, I focused on two systems. The mountain pine beetle, which you can see there is that beetle in the middle, and Rhagoletes flies. So that's a Rhagoletes fly there. Doing a combination of population genetics and starting my work in evolutionary and developmental genetics, which is where I see myself more now. But I also dabbled a bit in some barnacles and Drosophila work as well. Thankfully for me, my boss had similar thoughts on Kansas as I did. So I ended up moving when he changed jobs to the University of Colorado, which is a huge improvement over Kansas. If you've ever been to those two states, you'll understand why. So my largest project at Colorado was understanding how organisms adapt to seasonal variation. So as the seasons change, resource abundance waxes and wanes. And organisms living in seasonal environments must synchronize their growth and development and reproduction to favorable times of the year. And this helps them to take advantage of seasonally available resources. And a key question in evolutionary and ecological physiology centers on the regulation of phenology, which is to say, how do internal processes mark time to set the timing of key life history events in regards to the season, such as juvenile to adult transitions or the onset of reproduction? How does an insect know to emerge post-winter and start breeding? How does a plant know when to start preparing itself for winter? And a follow-up question to this is asking, how does variation in these processes contribute to adaptive evolution, which is often exceptionally rapid to track changing environments, for example, under climate change or during human-mediated biological invasions? And today, we're going to look at the timing of diapause. So insects in particular use diapause to time their development to seasonally available resources. And diapause is a hormonally controlled seasonal response to environmental cues that results in developmental arrest. And it enables insects to avoid unfavorable conditions. So we often think about this in terms of winter, but an unfavorable condition could also occur during summer. And insects are pretty neat. They can diapause as eggs, pupa, adult, all life stages, but only once in a life cycle. And during diapause, development, metabolic rate, cell proliferation are all suppressed until termination is triggered via environmental cues, which we can think of as something like light or temperature. And so this work focus is on Rhagoletes pulmonella. So pulmonella is a tephritic fly native to North America, and it's a textbook example of a species undergoing sympatric speciation, which is where a speciation event occurs in the same geographic region as the ancestral population lives. So in America, the native host for pulmonella is the hawthorn tree, and its general life cycle is this. You have an egg, which are laid in the fruit, in the hawthorn fruit, which they infest as larvae before emerging as a pupa, and they diapause through winter in the ground as a pupa, and then diapause is terminated somewhere near the end of winter, and eclosion into an adult quickly follows the termination of diapause. So when Europeans came to North America, they introduced apple trees, which pulmonella promptly started infesting. So we have what's known as two host races, apple and haw, and they're characterized by differences in their diapause phenotype. So you can see in this graph here, the apple race, which is the dashed line, it emerges just before the apple tree's fruit, and then the hawthorn race emerges just before the hawthorn tree fruits. And so this difference in the timing is about a 3-week difference, and it times into the fact that apple trees fruit earlier than hawthorn trees. And these trees occur right next to each other. These 5 populations still undergo extensive gene flow while maintaining this 3-week difference. And so the interesting thing here is that apple trees have only been present in North America for a few hundred years. So this is a rapid evolutionary change to exploit a new resource. And so what we want to know is, firstly, we just want to know how they know when to terminate diapause. How do they measure time so accurately that they emerge just before their fruit is available? And secondly, we want to know, how do apple flies rapidly evolve to shift their diapause timing 3 weeks earlier? And to answer this question today, we're going to look at 2 bits of data, mRNA sequencing and pooled genome sequencing. So mRNA data can give us an overview of the genes expressed at any one point in time, while pooled genome resequencing is an efficient way to assess where in the genome selection is acting. And so, but to set us up, I'm going to talk about some previous work to start with. So some previous work looked at transcriptomic differences at the end of winter. So samples were overwintered for 20 weeks, which in this fly is really great because you just stick them in the fridge for 20 weeks. And then they were sampled just before they were taken out of the fridge at 0 hours, and then 24 hours and 48 hours post-winter. And the question they were asking was whether the genes were differentially expressed between the host before coming out of winter, so they were already different by the time we got to the end of winter, or whether the differences between the hosts occurred post-winter. And what they found was that they were already different by the time they took them out of winter. And this wasn't really the expected result because they're not meant to be doing anything during diapause. Development is suppressed, but it implies that the difference between the two host races must occur during the diapause stage. So the first data set I'm going to talk about is a transcriptomic experiment to look at diapause changes during winter. And so we have flies, apple and hoarflies, and they're overwintered for 6 months, aka put in the fridge for 6 months, and we sample them every month from 2 to 6 months. And we do the same for both host races. And we are looking at the RNA expressed in the neural tissue. So this is a picture of a fly brain. They're about as fun to dissect as you can imagine. And we're interested in two main questions. The first question is, what is differentially expressed across time to see what's happening during diapause? Because it doesn't appear morphologically that there's any differences during diapause. But the previous results suggest they are. So we firstly want to know what they're actually doing during diapause. And secondly, we want to know what the differences between the two host races, as indicated by the arrows here. And can those differences explain that difference in diapause termination phenology that we see in the wild? And in this case, our hypothesis is that apple flies might be sort of more advanced compared to hoarflies. So they're a little bit ahead, and that allows them to move 3 weeks earlier in their termination timing. And so as we collected those RNAseq samples, we also tested how long each host race took to emerge from diapause if we terminated winter early, i.e. if we took them out of the fridge, how long did it take for them to eclose, which is when they emerge as an adult. And diapause termination in this fly is closely followed by adult emergence. And you can see from the results that they're concurrent with what we see in the previous work. Even by 2 months, there's a difference in the days to eclosion between the two host races. So there's something different about the way in which these two host races diapause. And you can also see that when we measured brain morphology from 2 months to 6 months, and there was absolutely no difference in the gross brain morphology between 2 months and 6 months. And when we look at the RNA-seq, broadly you can see there isn't any striking pattern. They don't overly cluster together, but this is what we expected. We're studying something so subtle it has no morphological change. We can't tell the difference between a 2-month fly brain and a 6-month fly brain. So we're not expecting drastic shifts in RNA-seq differential expression. We're expecting subtle shifts in gene expression through diapause. And that's what we get. So to answer our first question of what's happening during diapause, we looked at differential expression across the time series within each host. And here you can see the expression change across time, both the apple here and the whore to the right. And you can see about half the genes go up and about half the genes go down. And as you move from 3 months to 6 months, the colors get stronger, meaning that the expression difference gets greater. And this pattern is very consistent between the hosts. What goes up in apple tends to go up in whore and vice versa. And this makes sense. They're so closely related and they're still undergoing gene flow. They should be broadly very similar. And if we cluster those and plot those out, you can see broadly we have the same pattern across the two hosts. And that pattern is a linear pattern of increasing or decreasing expression, which wasn't what we expected to find. And these clusters are enriched for things like transcription regulation, cell cycle, differentiation, neurogenesis, EGF back domain, Wnt signaling. This shows that they are developing during diapause. It's just really slow. So in these flies, it appears diapause is not a cessation of development, but a slowing of development. And if we look more closely at some of those genes that are differentially expressed, we can see that they're definitely developing to some degree based on the types of genes that are expressed. We can see expression in the Wnt signaling pathway, which is involved in embryonic development. And we can also see changes in genes involved in cell fate and pruning. all confirming that they are developing, be it so very slowly that we can't measure that development on a gross morphological scale, but we can see it with RNA sequencing. So to sum up, broadly speaking, our results suggest that diapause duration in Pomonella is affected by the rate of development through the pupal developmental stage that has evolved an exaggerated expanded duration relative to the ancestral state of non-diapause pupal development common across higher flies. And then to answer the next question, which was whether expression trajectories during diapause diverge between the two host races to explain that 3-week shift with apple flies emerging earlier so that they can infest an apple tree. We looked for things that were significantly differentially expressed between host races at each month. So no longer looking along that time series as we were before, we're now looking between each month. So 2 months apple to 2 months fall, what are the differences? And you can see here that there's a whole lot of differential expression at month 2 and 5, with very little in the middle. And if we plot those out, you can see that hourglass pattern of expression differences between apple and whore host races across the developmental period, with high divergence early and late in diapause and nearly undetectable divergence in the middle. And so what we think is happening is that the duration of diapause may be sensitive to changes in gene expression during the diapause termination process, for which in this species is quickly followed by adult emergence. And this affects the seasonal timing in nature. Although we don't disagree that there is the potential for diapause initiation, the earliest phase, to also play a role in diapause duration. And we look at those differences between hosts, we can see a lot of differential expression differences between the hosts in the OXPHOS proteins that are involved in energy use and metabolism, ribosomal proteins that provide a signature of active translation, and core nucleosome proteins whose transcripts are generally only abundant during active cell division, all of which show an upregulation in apple at 5 months, suggesting that they are sort of developmentally ahead of the whoreflies. And we think this reflects an earlier potentiation in apple flies, whereby the chilling requirements necessary for diapause completion are probably surpassed by 5 months in apples. It's slightly different to being ready to terminate diapause. In contrast, in the whoreflies, we don't really see the uptick until 6 months. So they are about a month out, which times in with the 3-week shift. And the other thing we looked at was the influence of hub genes. So rapid evolution of a trait is often facilitated by the evolution of genes that serve as hubs in transcriptional networks, whereby changes in these hub genes are correlated in transcriptional pattern changes in many genes connected to the hub gene. And when we looked at the influence of hub genes in the evolution of diapause timing in the flies, we saw that yes, if you compared genes that were just differentially expressed across time, which is this random sample here, to those differentially expressed across time and between host races as well, so one's potentially relating to that difference between apple and whore, you can see that by the joining of the dots that there is more connectivity in the genes associated between host races. And this hub gene nature of the transcriptional changes may explain why there's been such rapid changes in the development within these flies, because it's only been a few hundred years since Europeans introduced apple trees. And so the second way we looked at the evolution of this trait was using whole genome resequencing to understand the genomic architecture of the trait. So this is a distribution of the flies, like I showed before. As we now know, apple flies emerge earlier than hawthorn flies, and they sort of have a normal distribution of eclosion timing. And so what we did for each host race is we took both bulk samples of earlier closures and late closures, and then we compared, we also took a bulk sample of a random distribution of closures across the curve. And what we did for each what we did then was we compared early flies in the apple and the haw and late flies from both host races to identify SNPs across the genome that are associated with diapause timing. And unsurprising, given the RNAseq data, we found that eclosion timing and host race differences are linked to a lot of SNPs. We found over 50,000 SNPs associated with emergence timing in both host races. And they're largely sit in regions that suggest dysregulation of genes, which means they're likely acting to regulate neighboring genes and not changing the protein structure itself. And you can see that in this chart with the overrepresentation of SNPs in introns, 3' regions and such. Just note that that's not a linear scale on the side there. And we have over 14,000 SNPs associated with host race differences themselves, and around 450 are associated with emergence timing and host race differences. And if we look at what they do, we see that they're involved in very similar things to what we've seen before in the RNA-seq, things like Wnt signaling, transcription regulation, and embryonic morphogenesis. And the position of these SNPs associated with emergence timing is quite interesting. And a cool discovery, we found that the SNPs associated with emergence timing are clustered on genome inversions. So you can see that there is an overrepresentation of SNPs in, so this is low, medium to high linkage groups. And there's an overrepresentation of SNPs in medium and high linkage group regions on chromosome 1 and chromosome 3. They only have 5 chromosomes, and these are associated with inversions. So inversions are just, they're a chromosome rearrangement where the chromosome kind of breaks and does a 180 and rejoins, and they have a number of different impacts, but one of the things they do is they reduce recombination. So you end up with areas of very high linkage where the regions of the genome are inherited modularly and it's not recombined. And genome modularity may help facilitate genomic responses to selection along multiple ecological axes and taxa prone to ecological speciation with gene flow. So the Pomonella genome has high levels of standing variation, and adaptive eclosion timing allelic combinations seem to be protected from recombination by linkage. And this type of genomic architecture may actually help facilitate the rapid evolution we see in these flies. They really commonly speciate onto new host species, and their genome seems almost sort of set up to do that. And the final question in this project that we asked is if general developmental processes affect diapause duration in Pomonella, we would predict that similar sets of genes may affect other phenotypes related to the development rate. And so we compared our flies to a study on Drosophila that used 600 generations of selection to develop an accelerated development line that would develop about 20% faster than control flies. And when we applied the same analysis that Burke et al. Used on our flies, which are really two disparate phenotypes, united only in their rate of development, we found that there was a significant overlap in the genes associated with development time in Drosophila and the genes associated with adult emergence in Pomonella, meaning that the development time in Pomonella seems to be determined by similar sets of genes that influence the rate of development in Melanogaster, which is to say that the rate of diapause development may be controlled by the same pathways that control general developmental processes. And finally, on this topic, I'm just going to mention that we're doing we have a comparative transcriptomic data set for the cherry fruit fly from Greece, which is another Ragelides fly, but obviously this one infests cherries. But in this case, instead of host differences, it's differences in diapause timing in allopatric populations that are separated by altitude. And you can see in this MDS plot, in this species, in contrast to Pomonella, we have a lot of differences between the two populations, which makes sense as they're not undergoing gene flow like Pomonella are. But we're currently comparing the results from Pomonella to Sarossi. Now, I'm not finished talking, but I'm just finished this section, so I'd just like to thank my collaborators, as there's clearly a large group of us working on this. I didn't do it all on my own, so thanks to them. And for the second half of this presentation, I decided to throw in some of the work that I'm doing now. So I moved back home to Otago to work with Neil Gemmell. There he is, doing some science, and Rabia Puon on parasites. And this has continued now, so I'm a research fellow, and I try to work on parasites, but I also work on a bunch of other things. So I dabble a bit in weevils and sea urchins and lamprey and biomonitoring. But today I'm going to present about parasites. And as I haven't had time to write something new for GA, I'm just going to give a talk that I did recently to a non-genetics conference. And I'm not even going to try and find the connection to my previous work, other than it's about insects, development, and transcriptomics. I tried to sneak in a few more genetic sides, but it's pretty light and airy. And in lieu of lots of genetics, I'm going to promise you some fun videos. So yeah, complete topic switch here. But parasites routinely manipulate their hosts to enhance their own transmission or survival. And this is known as extended phenotype, where the genes of one organism create a phenotype in another organism. There are many examples of extended phenotypes that range in subtle changes in feeding behavior to truly bizarre behavioral and physical changes. So there's 3 well-known examples. This is a wasp that manipulates its host into becoming a bodyguard. So the wasp lays its egg into a ladybird, which develops inside the host until it emerges and pupates. And while it's pupating outside of the host, the ladybird is manipulated to remain frozen on top of the pupa, guarding it. And this is an example here of a snail, a flatworm that manipulates its snail host to appear as caterpillars. So its eyes become colorful and pulsating, and this facilitates the parasite moving to its next host, which is a bird. So it not only makes its eyes colorful and pulsating, it blinds it at the same time. So it comes out during the day to expose it to bird predators. And finally, this is a nematode that manipulates its host to appear like a berry. So the nematode manipulates the ant to develop a bright red abdomen, which the ant holds in an elevated position while it becomes sluggish and slow. And this helps presumably move the nematode into its final host, which is also a bird, by sort of turning the ant into a berry. And so we're interested in parasites that manipulate their host to seek out and drown themselves in water. This enables the worms to emerge into a nice environment to reproduce. And this bizarre manipulation is induced by 2 phylums of parasitic worms, Nematomorpha, which is known as a hairworm. You can see here coming out of a wetter, and nematode, which manipulates its earwig host into water. So in this case, we have this fascinating manipulation being caused by 2 very different worms. And what it looks like is, hopefully this video will work. This is a tree wetter. It's doing something unusual. It's climbing through water. And that thing coming out the back of it is not a tail. That is a worm. So you can see they get really large. They start off as these sort of microscopic cysts, and they end up in these enormous worms. And they sit outside of the digestive tract in their hemolymph. But at this point, because they're so large, they kind of, all the reproductive organs and the digestive tract and stuff is greatly reduced inside the insect hosts. They're effectively sterilized. And so that was the wetter system. Today I'm going to start by talking about the earwig and the nematode system. It's a reasonably straightforward life cycle. There's only one host, the earwig. The nematode eggs are laid on plants. The earwigs consume the eggs, and the worm grows inside the host until it's ready to leave the host when it forces itself to drown in water, where the nematode mates before continuing its life cycle. And when I say they grow inside the host, I really mean it. They are absolutely packed full of worms by the time the worm is ready to emerge. And this is another view of that. So this is a micro CT, and you can just see how packed in that worm is. It's basically just an earwig shell with worm inside. So all the internal organs are sort of greatly reduced at this point. So how does a worm manipulate their terrestrial host to drown themselves in water? Our observations in the lab show that infected insects change their behavior quite dramatically at the final stages of infection, rather than normal insect behavior. So like in wetter, they kind of move around and burst in the arena. So they kind of go, and then they kind of still for a while, and then move again, and they're kind of still for a while. Heavily infected, where do they start moving into this sort of continuous drunk manner around their arena, and they become quite unresponsive to outside stimuli like sound. So to understand what's happening to the host, we're looking at gene expression changes in both the host and parasite. So profiling the transcriptome is the most efficient way to acquire a comprehensive snapshot of both the parasite and the host physiological state. And this allows us to understand the host physiology, how the host physiology changes so much during infection that a terrestrial insect is driven to drown itself in water. And we have transcriptomic data from earwigs' heads and their corresponding worms. We have data from uninfected, infected with small, medium, and large worms. And we also have the corresponding worm data. I'm just going to briefly talk about the head data today, though. So as we move through infection, we might see an array of transcription changes. As you saw before, these worms take up most of the body cavity, and by the end of infection, and as such, there's going to be some pretty drastic physiological changes that are not associated with the manipulation, but rather the infection itself. But we can narrow down our hunt for manipulation candidates by focusing on transcripts for which the pattern fits the behavioral trend we've observed. Through infection, we might see transcripts that continue to become more and more differentially expressed as the infection progresses and the toll becomes worse and worse. We also might see transcripts that reach a peak or a low and maintain that during infection. Others could have quite varied responses, but the ones that we're really interested in are these ones. Transcripts that are either dramatically increasing or decreasing at the final stages of infection, which is where we're seeing that behavioral manipulation. There's no benefit to the parasite if it induces the manipulation too early. And what we found is that through infection, there's around 9,000 transcripts that are differentially expressed. There's more upregulated than downregulated through infection, but there's a group of downregulated transcripts that you can see are severely downregulated during the final stages of infection. And if we cluster these by trend, irrespective of sign here, you can see that there's two sets that really move in the final stages of infection, which is the pattern of change we're interested in. And if we look at those two gene clusters, we can see that they're comprised by a lot of transcripts, which isn't surprising. It is difficult, but it does make it a little more difficult to figure out what's the actual manipulation and what's just a response to almost a sort of catastrophic infection at this point. But if we look at the broad processes associated with these trends for a gene ontology analysis, we can see in this first cluster is associated with things like neurotransmitter secretion, visual perception, photoreceptor cell axon guidance, olfactory learning, locomotory rhythm, short-term memory, and physical mediated transport. And the second cluster is associated with things like neurotransmitter secretion, methylation, along with immune responses. And a couple of those are quite interesting. Based on the behavioral observations, we know we're looking for some key changes in locomotory behavior of the earwigs. And we saw that locomotory rhythm was an enriched Go term. If we look at some of those genes associated with locomotory behavior in these clusters, what's really interesting is that there's some really heavily downregulated transcripts that have key roles in insect locomotion. And knockout studies of these result in abnormal locomotory behavior. That's similar to what we see in infections. For instance, DAP160 knockouts have large decreases in locomotory activity, while unfulfilled and AC3 knockouts develop erratic movements. And trippy knockouts lose fine motor control. And two of these you'll see also not only associated with locomotory differences, but they're also associated with sensory changes outside of locomotion. Because it's not just a change in movement, it's attraction and loss of aversion to water, which we think is driven by changes in the sensory pathway, which were also enriched in our Gemmell analysis. So if we look at changes of genes associated with sensory pathways, we see a lot of changes in regards to visual reception in the host, along with changes in chemical and olfactory and mechanical perception. We think this and the locomotory changes explain why they become responsive to outside stimuli in the final stages of infection. They sort of simply have limited control at that point. And so the visual changes are quite interesting. One hypothesis to explain the attraction to water is that visual perception changes so that they become attracted to light, which in the case of a nocturnal insect could often be the moon reflecting off the water, similar to the way hatchling turtles are attracted to light due to attracted to water due to light reflection. And our results suggest that this actually may be the way it acts in the nematodes as well, that they're not attracted to water per se, but they are attracted to the light coming off the water. And given the changes we see in the locomotory genes, they probably become trapped and unable to remove themselves from the water once they're in it. But what we're currently focused on doing is developing reference genomes for both hosts and parasites in the hope that we can get better gene models and hopefully better annotation, which will help the differential expression analysis, because we get a lot of things that we can't annotate. And some of them look like they're quite interesting in their sort of trend, but we don't know exactly what they are. So, and we can also, once we have the genomes, we can also do things like genome comparisons for convergent evolution and horizontal gene transfer. And so we're doing this for both sets of hosts and parasites. Upendra is a PhD student who's working on the Earwig and the nematode data. He's been assembling the genomes for both of these, and they're looking pretty good. The Earwig genome has an estimated size of 1 gig, and the current assembly using nanopore and 10x is around 1.4. And the Busco score, which gives an idea of how complete the genome is based on the presence absence of a conserved set of genes, is around 85%. So most of what should be there is there. It is in around 100,000 contigs at the moment. The nematode genome has an estimated size of around 330, and the current assembly is at about 580, spread across 5,000 scaffolds, with a pretty good Busco score of over 90%. I'm in charge of the Weta and the hairworm. I'm going to come back to Weta and why I made a mistake doing Weta there. The hairworm's pretty interesting. There's no publicly available genome for this entire phylum, so we have no idea what to expect. We think the estimated size is around 150, and the nanopore and 10x reads have assembled to around 200 on less than 10,000 contigs. The Busco score is only 77%, but I'm still working on finishing this one off, and there's very little to relate it to because there's no other genomes for the entire phylum. So the Weta is a problem. So I knew going in from my previous work on Aotearoa that Alphopterans suffer from genome gigantism. I worked on grasshoppers in my honors, and they're terrible to work on for genetics. And I, for some reason, allowed myself hope when I saw on the genome size site that there was one representative for Phytophoridae, which is where cave Weta sit, and it had a very reasonable genome size estimate of 1.5 gigs. And then when we ran Supernova on the 10x data, it struggled through and said that the genome size was over 4.5 gigs. And it just totally fell over. We don't have the read depth, in addition to 10x seeming to struggle with insect genomes. And then I subsequently was Googling around online, and I found 2 other people that have done genome size estimates on Phytophoridae. One person was trying to assemble this guy and learned that it had a genome size of around 10 gigs, and this other one was discovered to have 1 over 5 gigs. So I don't know how big this cave Weta is. We're currently doing a flow cytometry run along with some more short read sequencing to try and get a better handle on how big it is and whether it will even be possible to assemble it. Because we've been focusing on the Weta hairworm system for the past wee while, because we wanted to get a system up as a lab model. So this is the Weta hairworm system, and it's more malleable for the lab as a lab model as it has an intermediate host. So in this system, the hairworm lays eggs in the stream, they develop into free-living larvae, which are consumed by an intermediate host, which is just a freshwater invertebrate of some type, so mayflies, caddisflies, and the parasite forms a cyst inside this host and doesn't do anything to this host. It just hangs out there until the host leaves the stream as a natural part of its life cycle, where it dies on land. And the Weta scavenge the dead intermediate hosts, get infected by the hairworm, where it grows into these massive sizes before manipulating the wetter into the stream. And so what we should be able to do is take these intermediate hosts and feed them to wetter in the lab. And that's much better for this type of work, as we can control the conditions and the infections. People do it overseas all the time. It works really well. We spent quite a long time trying to infect wetter in New Zealand, and we've tried to infect crickets and we've tried to infect cockroaches. And we've also had problems with most of our largest experiment on this being interrupted by COVID lockdown. So at this point, we sort of decided, okay, it's not working well here. Let's collaborate with someone that's doing it overseas. And that's a reason I like science, is that problems can sometimes create opportunities to collaborate. So we're currently working with a colleague in New Mexico in the States who's doing the infections for us. And we're also developing a further collaboration with a researcher at the University of Bath in the UK who looks at secreted small RNAs and parasites. COVID's not been the best time to start an international collaboration, but it's just been kind of nice because it can feel a little bit isolated sometimes in New Zealand. So it's been nice to start a collaboration during COVID. And this is a general infection cycle that Ben has in his lab. So it's a really, really good system for the lab. It's a sharp 28-day cycle. You infect on day 0, the worm emerges on day 28. So it's a massive growth in a short period of time. And so what we're doing is we're collecting for RNAseq of the wetter, not the wetter in this case, it's a cricket, and the worm at day 15, day 20, day 25, day before infection, day before exposure to water, the night before exposure to water, the day before exposure to water, and then during exposure to water. And the nice thing about this system is that the cricket genome is already sequenced, it's in the final stages, and the hairworm sequencing is also underway. And the second project we're working on with our international collaborator is on this. So this is a wetter bottom. You can see the ovipositor here and a couple of legs. And you can see here that there is 4 worms emerging from this wetter. So multi or dual infections are really common. About 30% of all infections have more than one worm. But what's interesting is that they have worms of different ages. So this means if the older worms are driving the insects to death in water, they're also killing the juvenile worms. And you can see in this picture, this is a juvenile that was undeveloped inside the host. So they don't turn that nice brown color until they're an adult. And so those 4 adult worms, by driving that host into water, also were killing this undeveloped worm. So you have a conflict not only between the parasite and the host, but between the parasites within a host. And if we go and look at worms in the wild, we can see that sometimes we would predict they would collaborate as they're similar sizes. So in these first two examples, we have a size ratio of 1 to 1. And so we would predict that these worms would get along quite well with each other because they're at a similar developmental stage, and they should reach emergence at around the same time and collaborate to drive the host into water. And so in these cases, we think that the host likely acquired the worms around the same time. But in these other cases, we have worms that have quite a different ratio. Some of them are half the size of the other worms. And this suggests that the host acquired worms at different times, and this age difference put the worms in direct conflict with one another. If these older worms kill the host, they also kill the undeveloped worms. And the second factor is that the relationship between these worms varies from full sieves up here to unrelated. And so you could see a situation where they are different sizes, but the worms are siblings, so maybe kin selection might favor that they collaborate with one another despite their size difference. And so with Ben's crickets, what we will do after he's finished this experiment is we're going to do a dual infection setup. And so we're going to infect the crickets once, and then we'll infect the crickets again at a different, a couple of weeks later. And we'll look at the expression differences in the host and the worms to see how a dual infection impacts the host manipulation. And if you check back next year, hopefully I will have a nice presentation that I can give on that data. So thanks to my collaborators with this project. And I do have another video I can show you here. So this is a guy, cave wetter in the lab. And you can see he's, again, he's doing something weird for a wetter. He's in the water and he kind of puts his butt in the water. It almost looks like he's sort of ovipositing. And in a moment, we just have to wait for it, the worms start coming out. So I think this guy might have had 3 or 4 worms in him. Yeah, so there's the first worm there. And there's another couple there as well. Interestingly, the worms that Ben uses in the States, they emerge a lot quicker in those crickets than the worms emerge from what we've seen in the wetter. I don't really know why some worms would emerge quicker than others, but ours apparently are slow to emerge compared to his. But yeah, you can see that poor wetter had a ridiculous number of worms inside him. Yeah, so thank you for listening.
I'd just like to thank Eddy again for a fascinating talk. Again, lots strong on the insect, so that makes me happy. But also thanks for, as you said, stepping in at short notice to fill in the slot. And we promise to give you a lot more warning next time, because there's some really fascinating science here. So thanks very much, and I'll do some more clapping, and I think everyone else is. As I say, if anyone wants to be on our mailing list, please let us know, and we'll see you again in a couple of weeks. So thank you very much, and goodbye.